Okay, hello everyone. We're on Friday. It's September 25th at about 2:40 in the afternoon. Today I'm joined by a couple of folks. First of all, Paula Harvey, who is one of my colleagues at the Tejon Conservancy. I'm sure you will hear from her later. She is the person who was the mastermind behind the biology lab notebook format that you've been working on a lot so far. And so I'm sure you'll hear from her a little bit later on during this recording. We're also joined by Scott Beering, who's a postdoctoral scholar at UC Berkeley and Dr. Eva Harris's lab. He's a virologist who specializes in the coronavirus. He's studying it I believe, almost exclusively right now. And so he is going to give a presentation on viruses and going specifically toward coronavirus. And then thanks to those of you who submitted questions in the Ask a Virologist assignment. I have your questions in a PowerPoint after Dr. Beering is done with his presentation. We will open up that presentation and allow you to ask your questions in your recordings, and some of them he may have already covered, others not. So anyway, with that, Dr. Beering, thank you again for being on the class.
Thank you so much, Mitchell, for having me. And I'm kind of excited to share my work with you today about some of the stuff we've done with SARS coronavirus 2, the causative agent of COVID-19 coronavirus disease 2019. So these slides that I prepared it's kind of an interesting time because they're from April. I prepared them in April. And since April, a lot of things have changed. So I'll kind of update as we go, like what we know about it. So, I mean, on this slide, for example, this was kind of like a prominent case map of, where in the US different COVID-19 cases were reported. And at this point, this was back when New York City was being hit really hard, and you can kind of see this explosion of cases here. But California, more or less, was still kind of like low. Since then, California is one of the worst places. LA and San Francisco have many, many cases, and it's still a very serious issue that's ongoing. And as Mitchell mentioned, I'm a virologist, which means I study viruses. There's a lot of different types of virologists. There are epidemiologists who study kind of more on the public health level, how it affects populations, what trends you see in basically in the real world that lead to outbreaks of viruses and stuff like that. I'm a molecular virologist, which means that I study viruses in the lab. I try to understand how they cause disease, how they infect cells, and how you can prevent this. Things like vaccines and drugs, discovery for antivirals there. Also, as Mitchell mentioned, I'm in the laboratory of Dr. Eva Harris, and normally we don't even study coronaviruses. Normally we study viruses called flaviviruses, so like dengue virus and Zika virus, which you may have heard of. But since shelter in place has happened, essentially kind of all virologists have been kind of like called to arms in a way, and we've all been kind of focused on finding quick translational ways that we can treat COVID-19 and lower basically the chances of death once people report to the hospital. So I'll kind of go through this with the context of what is essentially an emerging viral infection, what is SARS coronavirus 2, and what are some local and global responses that have happened since shelter in place has started and kind of where we are right now. So the concept of emerging viral diseases is one that's been around for a long time. And I think this outbreak with SARS coronavirus 2 has essentially brought it to the forefront, more or less guaranteeing that people will pay attention to the topic of emerging diseases, hopefully forever. But there's always been this potential for these types of outbreaks. And I'm sure you've all heard of SARS coronavirus 1, which is highly, highly related to SARS coronavirus 2. So in a way, we kind of had a warning in the early 2000s when SARS coronavirus 1 basically had an outbreak in China. Now, this virus was not as easily transmissible as SARS coronavirus 2, so it kind of dwindled out. But I would say overall, we didn't learn our lesson. We did some research on SARS coronavirus 1, but I would say interest in the virus kind of waned because one, the virus is too dangerous to study in a normal lab, so it required special barrier to study it. And two, there are a lot of other viruses that outbreak all over the place. So I guess the question is, what do you study when? But we'll kind of talk about a little bit of like how can you predict an emerging virus? What is going to be the next pandemic? Because certainly this is going to be a trend that we see in the future. SARS coronavirus 2 is really bad, but I'm sure that there will be other viruses in the future that will also emerge into the human population as we continue to expand. Such viruses, like we've heard about Ebola virus a lot, and that has emerged and reemerged, which means that it's not a new virus, it's just come into a new set of hosts and has an outbreak. But things like Ebola don't spread as well. What's really bad about SARS coronavirus 2 is that it's a respiratory virus. It transmits very easily from person to person. So going a little bit about this kind of emergence of viruses, a lot of these viruses are zoonotic, which means that they can be transmitted back and forth between people and animals. And many viruses that have what you would call a reservoir, so either an insect reservoir or a mammalian reservoir, these viruses can be very difficult to predict and to predict how they will mutate. The reason for this is that maybe we can monitor patients when they go to the hospital, humans, but we're not super aware of what's going on when these viruses are in their animal vector, meaning they can be mutating and they can be changing here. And as we change the environment, as we move into new environments through deforestation that we haven't seen before, we may encounter these viruses that have the potential to infect humans. So a general trend, so I guess I kind of said this before and I'll kind of emphasize over and over that a lot of times when viruses emerge, they're highly virulent, but they don't easily transmit between humans. The virulence of the virus usually refers to the fact that it's not adapted to the human population. A common misconception is that viruses want to kill you, that it's beneficial for the virus to cause severe disease. Sometimes this can be the case, but usually not. The virus's goal is to just propagate itself, to continue to exist. And if it can do that and keep you healthy, the chances are that it can infect you longer. If it ends up killing you, then it can't propagate for as long as it wants to. So these viruses emerge, they're usually highly virulent because they're not adapted to a population. And as time goes on, they may become less virulent. So I'll touch on that topic a little bit more later. So this is zoonoses with animals. I kind of mentioned this, that as the human population kind of expands and grows, we take up new environments. We do things like deforestation for economic purposes or for housing. We see new environments that we're not used to seeing. And through urbanization, travel, and things like commerce, we're able to spread viruses that we find in these places very, very quickly. So you can have a global pandemic very easily. We knew about SARS coronavirus 2 outbreaking in China months before it was announced as a global pandemic. And I guess it's kind of like a shame on us. We didn't respond fast enough. We assumed we had certain measures in place. A lot of the issue was that early cases were reported to be travel acquired, meaning that people who were in endemic areas like in China had a higher case of getting it. So domestically, we would say, okay, well, it's not something to worry about because it's probably not transmitting well between people to people. Which is not the case. After enough time, when it came here, it transmitted very rapidly. It transmits between households. I'm sure you've all heard of at least, friends or family, if not your own family, getting infected and spreading it very, very easily. So let's move a little bit into viruses, just kind of in general, and then we'll bounce back out to SARS coronavirus specifically. So I'm sure, as you're aware, viruses are kind of on that cusp of whether or not they're considered alive or not. Most biologists would not consider them alive by the standard rules of biology, which would be that an organism has to produce its own energy, that an organism has to essentially use its own machinery to do its life cycle. viruses are parasites in a sense that they require our cells to grow. They cannot produce their own energy. They use our ATP, they use our host machinery to replicate their genome. But viruses do encode their own polymerases, and they do evolve. They change and they adapt. So they do have some elements that we would consider living, but this is probably will always be a debate. You know, are viruses an early primordial form of life, or do they kind of like de-evolve to basically just the basics of what they need to propagate their genomic material? So at the core, a virus is either DNA or RNA, and this is the genetic material that encodes for a set of proteins that can do everything that the virus needs, basically setting up its replication niche inside of a human cell. This genomic material is protected by a protein capsid. DNA and RNA can be pretty fragile, so it has to be protected from the environment. And some viruses even have a lipid envelope that is taken from a cell that it infects. And they'll usually have proteins on top of this lipid envelope that help it to find the cells that it needs to find to infect. So yeah, that's essentially mostly on the structure. I would say that how you decontaminate yourself when you're trying to like not be infected by a virus depends on its makeup. As I mentioned, some viruses have this lipid envelope and some don't. Coronaviruses have a lipid envelope, which essentially means that they're a little bit less stable in the environment. Things like alcohol and ethanol can inactivate this envelope. But viruses that don't have this are less sensitive to this, and you need things like detergents like soap or even bleach to kill the virus. So coronavirus itself is not that stable on environments. So which is kind of a good thing. If somebody touches a surface and had droplets on their hands, chances are the virus is fairly unstable there and can't last very long. So I would say that if someone touched a surface and they were infected, and then they came, you came a couple hours later, there's probably no infectious virus there anymore, at least not enough to make you sick. There's many different families of virus that have different sizes, different rules. I would say that just like the general rules are that DNA viruses, they generally want to go into your nucleus to replicate, while RNA viruses generally replicate in your cytoplasm, which is where in your host cell you find DNA versus finding RNA. So it's a pretty simple rule. You find DNA in your nucleus, DNA viruses like to go to the nucleus. You find RNA in your cytoplasm, RNA viruses like to go there. Because I think I mentioned coronaviruses, where right here are RNA viruses, the lipid envelope, and they like to replicate in your cytoplasm, not in your nucleus. So other viruses that I'm sure you're aware of are retroviruses like HIV, filoviruses. This is the family that hosts Ebola virus. I'm sure you've all heard of herpes viruses, but there's a lot of different families of viruses that comes in all shapes and sizes. So today we'll talk a little bit more about coronaviruses. And this is what's interesting, that coronaviruses are not, it's not a new family of viruses. We're not just finding out about them. We've known about them for a long time. And before the emergence of SARS coronavirus 1, we really only knew about alpha coronaviruses, gamma coronaviruses, delta coronaviruses, these viruses that cause the common cold. So we never considered them to be that biomedically important. So the National Institutes of Health and the CDC will prioritize viruses that we should research based off of the availability of drugs and vaccines and whether or not it causes a serious disease. So coronaviruses cause a large proportion of virally induced colds. So you've heard of rhinoviruses, there are coronaviruses. And because they were so mild, we never really thought that they would have this pandemic potential. And we'll tell you a little bit more about why this is, but it's interesting. So SARS coronavirus 1 emerged in 2001, 2002. It was the first coronavirus that we discovered that caused significant morbidity and mortality in humans. And since then, in essentially a 20-year span, we have found MERS coronavirus, the Middle Eastern Respiratory Syndrome coronavirus, and now SARS coronavirus 2. Both SARS and MERS are more deadly than SARS coronavirus 2 in the sense that they cause a higher morbidity rate in a given patient. But SARS coronavirus 2 has killed far more people than these two viruses because it's more easily transmissible. And as far as the zoonosis goes, at this point in time, we're pretty certain that the closest ancestor of SARS coronavirus 2 originated in bats, and there probably is no intermediate host. It probably came right from bats to people. And how did this come to people? This could be explained by meat markets. It could be explained by people who live close to areas where infected bats are, and then the bat is defecating on something that we're interacting with and infecting them. So coronaviruses have a complicated zoonosis that's kind of highlighted here. So we'll talk and compare and contrast SARS-1, MERS, and CoV-2 as we move through the slides. Sorry, the slides are, there we go. So SARS coronavirus 1 and MERS coronavirus, two similar viruses that are highly related to SARS coronavirus 2. We'll talk about them a little bit. SARS coronavirus 1 has an intermediate host, this civet, which is kind of like a cat. It's a Chinese animal, and this was believed to emerge from bats because of meat markets, which is part of the reason we believe that SARS coronavirus 2 came from a reservoir in a similar fashion. MERS coronavirus emerged in Saudi Arabia, and its intermediate host is the camel. And in Saudi Arabia, camel meat is a staple, and that was people who farmed or worked or raised camels. That was believed to be how it transmitted. And then there were some cases, but mostly it was hospital acquired. Essentially, you needed to be around sick patients a lot. It didn't transmit as easily as SARS coronavirus 2 did. So if you compare these two viruses to, so this is an old strain, so we call it SARS coronavirus 2 now. At the beginning, we were calling it the novel coronavirus. And this happens a lot where when we're discovering something, people don't settle on a name yet. You need genetic information to know what you're dealing with. You need to kind of understand where it came from to come up with an appropriate name. So eventually we came up with SARS coronavirus 2, but at the beginning it kind of had a bunch of different names. So MERS and SARS-1 came first, so 2003 versus 2012. And the number of people they killed was pretty low relatively. So MERS and SARS-1 both, so I think SARS-1 had something like a 10 to 15% more mortality rate, while MERS has like a 30 to 40% mortality rate, which is pretty scary. But given that only 800 people essentially have been killed by the virus, the infection rates were pretty low. And we still have cases of MERS coronavirus today, but they're a little bit more rare and they don't transmit well. SARS coronavirus 1, we have not seen this essentially in the wild since it emerged and went away. And this is part of the reason we thought it wasn't a threat. But the virus emerged, infected some people, and then it kind of cleared itself. So we kind of thought, okay, maybe there's not a lot of capacity for this virus to transmit well. This novel coronavirus, though, that it has a lot of interesting traits that make it transmit really well, and we'll go through that a little bit more as well. Trying to advance this slide forward. So we talked about bats a little bit. I won't cover this as much. This is talking about the same thing. We're talking about the morbidity mortality rate. So yeah, this is redundant information. And then if we're comparing the epidemiology and public health of these viruses, this is kind of a central thing that we're getting into now. A number that is an important number in epidemiology and public health is this value right here, referred to as R0. And what R0 means is, on average, the number of people who are infected for any given case of the virus. So if one person is infected with flu, for example, on average, 1.3 other people are infected from this one person. This person transmits to 1.3 other people, and there's no such thing as 0.3 of a person, right? So this is kind of a statistical average. So flu itself is not highly transmissible. It is, but not as much as some other viruses. SARS-1 was highly airborne and permissive, but it burned itself out too fast, and it had a couple other reasons why it didn't stick around, despite the fact that its R naught is fairly high. COVID-19, SARS coronavirus 2, the R naught is 2 to 2.5, which is pretty high, which means that it's able to infect a lot more people and say the flu is able to do. So in a given flu season, yes, you have people that are spreading it around, but in this pandemic, we have more people getting it per average. And then these numbers are all kind of like, being defined now. And I'm sure now we have, millions and millions of cases in the US even, and deaths in the world are approaching 1 million, I think. So cases increase every day. I have to pull new numbers every time I'm going to give a talk on it. So I guess differences between flu and COVID-19, and this is what happens. A question that I get a lot is everyone will say, okay, have I been infected with SARS coronavirus too? The virus itself can, it causes, most people who get it do not get severe disease. And a significant portion that get it are in fact asymptomatic, meaning they never even are aware that they can get it, but they can still shed. So the number of people who get severely sick it's fairly small given how many people can be infected. So the actual number of people who actually are infected are a lot lower. So I guess the point I was making was that people were saying, oh, I got a flu-like symptom maybe in December or in November. Do you think I got COVID-19? Well, it's possible. But we have done actually a lot of screening of people who were sick around this time, and most people did not have antibodies against SARS coronavirus 2, and antibodies would be a sign that they were previously infected. So just the basics of COVID-19 is you get the virus, you get infected, and if you're going to show symptoms, usually between 1 to 14 days is the incubation time, which is why the general rule for quarantine is that if you are quarantined for 14 days, you can be, and you're not sick at the end of that 14-day period, you can be fairly certain you're not infected. The main complications of COVID-19 are respiratory complications, so ARDS, so acute respiratory distress syndrome, where essentially you have hyperinflammation in your lung and you're not able to exchange oxygen into your blood. So most people who succumb, usually it's this cytokine storm that results in not being able to breathe. And prevention is self-isolation. So in common with flu is that you get fever, cough, and fatigue, which a lot of people get for all sorts of different things, not just the flu, for any type of cold. So it can be extraordinarily difficult to diagnose. And a lot of times, primary physicians won't test you unless your symptoms become more severe, because they just don't have the capacity to do that. So at the end of the day, these are the differences between these viruses. More people are symptomatic with flu than with COVID-19. COVID-19 does have a lot of asymptomatic cases. And as I was mentioning, the main symptoms are respiratory, and these are kind of like, these are the non-severe symptoms, and then severe cases, again, are this severe inflammation in your lung, which leads to lack of breathing. But as we've studied this virus more and more, we find that it's quite systemic. It doesn't just localize to your lung. It's viremic, meaning it gets into your blood, and it can replicate well in many other tissues, like your liver, your heart, your spleen, your kidney, your intestine. It can be passed fecal orally, which is very interesting for a respiratory virus. This virus itself can essentially go anywhere. And there's actually even evidence that it's sexually transmitted as well. We were reviewing a couple of papers on that. And I guess in a way, why does that matter? Why does it matter if a virus like COVID-19 is sexually transmitted, if it's respiratorily transmitted? But it does come to the point where who sheds it? How long do you shed it? And are there people who are totally asymptomatic and they're shedding it in one way versus another? So this is a constantly evolving situation. It's a new virus that has broad tropism, so we don't fully know all the answers to these questions. And all we can do is just collect data as we go and learn more and more about it. So as I mentioned before, we think now that there isn't an intermediate host. Originally, it was hypothesized that the pangolin may be an intermediate host, which kind of served like similar vector to the civet that I mentioned earlier for SARS coronavirus 1. But as I was mentioning, evolutionary biologists now have found that the closest ancestors to this virus can be found in bats, and not so much in pangolins. There are close ancestors in pangolins, and to be clear, all sorts of animals harbor many different viruses, and you can go and you can collect samples from animals and you can find sequences that look a lot like viruses that we interact with on a daily basis that cause human disease. And generally, when you're trying to figure out where a virus comes from, you ask, where is the virus that's most closely related to it? And that's usually where it comes from. Because as a virus gets into a new environment, it'll adapt, it'll mutate, it'll evolve to look different depending on that environment. So right now, the pandemic we're experiencing is mostly this cycle right here, from infected human to infected human. And this is why this is so bad, because it most likely came from bats and a zoonosis, but now it's maintaining in the human population. And we don't know too much about lasting immunity as well. There's good evidence that some people are well protected, but there's also been evidence of reinfection. So we don't know if people who get it once can get it again. There's evidence for both sides, honestly. So back on the point of why is COVID-19 SARS coronavirus 2 so bad compared to coronavirus 1, these viruses are very, very similar. And SARS coronavirus 1, in fact, has a higher morbidity rate. But why is SARS coronavirus 2, why did it change the face of our planet, essentially? Our society and everything that we consider normal has been rewritten because of this new virus. So these are kind of like a couple of major factors. One initial thing was that we weren't sure what the intermediate host was. For coronavirus 1, we know that it's the civet, and you can cull the chain, as in you can, like, if people are bringing in civets for pets or for meat, you can stop that and you can slaughter the animals that are normally used for meat and get rid of them. And originally it was like, oh, we didn't know what the spillover was for humans. But right now, I think it's pretty stable within the human population. And I think our own lack of submitting to things like basic social distancing can be partially to blame for why this isn't taken care of yet. A lot of epidemiologists will say that if you have a magic wand and you could make everybody just freeze in place for 14 days, this whole thing would burn through and it would just be gone. Whether or not that's the case is not for sure, but definitely the majority of the problem would be gone if that were if you could do that. And so this here is widespread community transmission is why SARS coronavirus 2 is worse than SARS coronavirus 1. So SARS coronavirus 1 did not transmit well between humans. And most of the time, this happened in hospitals where people were severely infected and shedding a lot of virus, and hospital workers were exposed to this. But given this, you could have higher, what they would call barrier nursing, or higher quarantining in a hospital. But coronavirus 2 spreads asymptomatically, which is the third point here. So people can be infected and they don't even know. I mean, to actually fully quarantine, you need to know that you're sick. If you don't know that you're sick, you may not change your behavior and you can be transmitting this. So that's the other reason why it's so bad. Moving past that, the virus itself, we'll talk a little bit about the structure, but I talked to you before that this is a virus. It has RNA, and it has an envelope, and it has a protein on the surface called spike. And this spike protein is important, and this is kind of important when it comes to vaccines. I won't talk about vaccines too much in this, but kind of what our hope as a society are is there's essentially three ways you can move past a pandemic. One is the virus burns itself out because it doesn't efficiently transmit, which was the case for SARS coronavirus 1. Two is everybody gets it and everybody becomes immune to it, and then you have what's called herd immunity. And you need something like 70% of the population to be immune before you can actually achieve good herd immunity, and people who are not immune would essentially be safe. And then you could have antivirals that could treat a given case, and even though maybe it's still moving through the population, people who get severely sick can be treated in a way that minimizes or completely prevents death. And then the other is vaccination. Each strategy has its pros and its cons, and I think generally speaking, it's well accepted that vaccination is the best preventative measure. I mean, I guess burning itself out would be best, but vaccination is important. And I'm sure we have, like, this isn't a class about vaccines, but the way vaccines work is you either put the whole virus in a weakened form or different proteins or pieces of the virus into a human body, and you let the human make an immune response against that protein. And for viruses, the most popular thing to make vaccines against are things on the surface that antibodies are going to see a lot. So this spike protein is the major component of most vaccines. And I guess I'll just give a little background. I didn't mention it earlier, but SARS coronavirus 2 is a coronavirus. And the reason coronaviruses have their name is because under microscopy and electron microscopy, these spikes look a little bit like a crown, and corona is Latin for crown. That is why coronaviruses have the name they do. So this spike protein is unique in that sense. When you're deciding on what the right strategy is to treat a virus, you essentially need to find drugs that block the ability of the virus to replicate. I guess I'll just kind of gloss over this, but the generalized life cycle of how SARS coronavirus 2 replicates in your cell, if this crude circle is your cell and this blue circle is the nucleus, the virus would approach the cell and it would use its spike protein to bind to the receptor on the surface of the cell. Once it binds to its receptor, and you may have heard of this protein, it's called ACE2, it gets inside of your cell. It usually does this in a organelle called an endosome. Once the virus gets into your cell, it releases its RNA into the cytoplasm, which is any area that's not the nucleus. And this RNA can then translate into proteins, into viral proteins. These viral proteins will then make the cell perfect for replication of the virus, to copy its RNA. And it does this in a structure called the replication complex. And it's really neat because viruses are able to make basically your cells their home. So they take all of your membranes and they make this perfect little house sheltered away from all other things that could harm it and just replicate like crazy. And in there, they will take their genome, they will make a lot of little copies. When the time is right, these will then get packaged into more viruses. And usually the result is your cell dies and these viruses can get out and they can go infect new cells. So if you block one of these steps from happening with a drug, that can either interact with a protein or block this process from happening, that's more or less how antivirals work. And actually I would give you an example. So there's a lot of vaccines that we have that completely prevent viral infection, but there are several viruses that we've never been able to completely successfully make a vaccine for. One example is HIV. But people who are infected with HIV now could be completely treated and more or less cured if they maintain a drug regimen. So you can actually live a healthy, successful life being infected with something like HIV, which used to be deadly and would kill most people that were infected with it. Hepatitis C virus is another example where pharmaceutical interventions can essentially make it more normal. So this would be somewhere in here you would want to block pharmaceutically. I'm going to skip this. This is an old number of how many people were infected in April. So at this point, the world had experienced 200,000 deaths, and we must be over 1 million now. I haven't looked for a couple of days, so this was still fairly early. These numbers have just jumped. At this case, we were comparing cities. New York totally has leveled out. It's really interesting. It has burned through New York, whether that's because they've achieved herd immunity or they've increased their measures on how to contain the infection. It's not completely clear, but New York was a major hotspot, and that is leveled out. I think on this list now, LA is probably the worst place. So LA County, Riverside County are some of the worst places in the country right now as far as coronavirus cases. This is another source that was listing deaths and cases in different countries. This is more from a country view. This was after we had surpassed China. So in the world right now, I think that the US and Brazil are probably the worst hit, but probably there's some European countries like the UK and Italy who may have a higher per capita death rate than us. But again, I haven't looked at it for a couple of weeks. I'm going to skip most of the pathophysiology, but I have a couple of slides about the disease that the virus causes in your lungs. We understand more about this now, but essentially your lungs are these little airbags. You breathe air into your bronchi, which then lead to these little air sacs called alveoli, which are lined by these epithelial cells. And these epithelial cells come really close to your blood vessels, which are lined by endothelial cells. And the idea is that your blood vessels need to have red blood cells that pick up oxygen, which then oxygenate your blood and allow you to make ATP in your entire body. So this layer right here has to be very thin, and it has to be, I guess, a complete barrier. So this is blood. You don't want blood seeping into your air sac, but you still want this layer to be really thin and able to have oxygen cross, right? So when you get infected, when you breathe in virus, the virus will then infect pneumocytes, epithelial cells that line your bronchi. We talked about this. The virus interacts with its receptor to get into the cell. I'll skip that. And when the virus infects your cell and it kills the cell and it shoots out more virus, these cells are going to produce things known as cytokines. Now, cytokines are little proteins that your immune system produce that essentially help respond to an infection. So cytokines will tell things like immune cells. Maybe you've heard of B cells and killer T cells, which will either kill virally infected cells or produce antibodies against the virus. But cytokines can also be a bad thing. They can actually cause pathology because they do result in tissue damage. So you need some cytokines to basically tell your body to respond and protect you from the virus. But if you keep making cytokines, it's going to cause tissue damage, which makes it hard to breathe. So I was mentioning these cytokines. These are a couple more. You have immune cells that come in to deal with the virus, to kill virally infected cells. But these cytokines cause permeability in your endothelial cells and cause this barrier that is needed for normal oxygen exchange to break down. This results in even more stuff coming from the blood into your alveoli, which causes kind of a self-amplification of more cytokine production and more tissue damage, which eventually just leads to scar tissue and your bronchi filling up with fluid, which is known as pulmonary edema, which just makes you essentially not be able to breathe. And the breakdown of this is what causes severe pathology and is what's known as ARDS, or acute respiratory distress syndrome. And then this virus, this is how it gets viremic, how it gets into your blood. It'll move through your blood vessel, and then it can get to other places where it wants to replicate. And at the end of the day, the virus wants to make a lot of itself so it can get out and infect a different host. It'd be better for the virus if it didn't cause this severe pathology, but more or less what makes people really sick isn't the virus so much as how we respond to the virus. That's why you see such a different outcome for different people infected. Some people are asymptomatic because their body produces the perfect immune response that totally kills the virus, gets rid of the virally infected cells, but then shuts itself off before it causes a lot of tissue damage. The people who get really sick, this usually happens actually after the virus goes. So the infection itself can be kind of separated into 3 stages. And the first stage is the viral response stage, or when you can even measure the virus. And this is only a couple of days, about day 1 to 4 after infected is how long you can actually detect virus. And this is kind of when you have this fever, cough, and fatigue and headache. And then into stage 2, usually the virus starts to go away. Your immune system starts to control the virus. So most people do this normally. And then after stage 2, you're supposed to turn off this inflammatory response. And the virus by this point is gone. And if you don't turn off this inflammatory response, you end up doing a lot of tissue damage and you end up hurting yourself. And that's the disease pathology. People die without having any virus, and the virus is already gone. It's basically your own immune system. I'm going to skip therapeutics. Therapeutics are interesting because I'll say that there's 2 arms of therapeutics. There's ones that directly target the virus, so antivirals, But those really, as from what I just told you, they really would only apply early. Once the virus is gone, the therapeutic doesn't do too much. So the other therapeutics you can take are things like anti-inflammatories. So one serious inflammatory molecule that I'll talk about is interleukin 6, IL-6. This causes pathology. So this drug is an antibody that blocks IL-6 from amplifying this immune response. So you can target either the virus or the inflammation. It's more or less two strategies. So what have we been doing? As Mitchell mentioned, I'm at UC Berkeley, and UC Berkeley itself was completely shut down from, I guess it was early April until we're still shut down now. We have come back to work about 25%. There's no students on campus. And most of the work that we're doing here is prioritized for COVID research, studying the community, studying transmission rates, doing drug discovery, and then basic research questions. So as I mentioned, I'm a molecular virologist. So I'm highly involved in taking the virus and trying to find new drugs that can be repurposed, and then trying to figure out how the virus works. There are other things that happen in Eva Harris's lab. One is we set up some early clinical testing. This was before hospitals had the infrastructure to test. This was something that the US did not do well. We didn't have the infrastructure to test as many people as needed to be tested. The viral test kits were rare, and the ones that we had weren't super reliable. So viral testing can be usually done by detecting either the RNA of the virus or the protein. So one thing that we did at UC Berkeley is we set up our own in-house, the assay is called an ELISA, and it detects viral proteins. So we did that in-house, and we could work with hospitals to do clinical testing. And also one thing that we actively do on campus is anyone who's on campus has to get tested once a week. So we have these little booths, we go up to the booth and we spit in a tube, and then they run a PCR, so they detect viral RNA and say whether or not we're infected. Another thing we've been doing is trying to understand how many people are asymptomatic, how many people are actually getting infected. Because if we want to understand herd immunity and how far or close we are to herd immunity, we need to understand how many people are actually infected. So one thing we started was a Bay Area asymptomatic cohort, and specifically East Bay. So San Francisco is across the Bay, and the East Bay would be like Berkeley and Oakland, and areas around there. So what we did was we mailed out thousands and thousands of flyers that invited people to enroll in our study. And the requirements for enrollment were not having a severe case of coronavirus, and not having more or less not having a confirmed case. If people had a confirmed case, we wanted them to enroll as a control study, essentially. When people were enrolled, we mailed them kits that gave them a tube to spit in and a little lancet to prick their finger and bleed on a piece of paper. Using that blood, we could look in their blood to see if they had antibodies against the virus. The spit, we could look for whether or not they had any virus in their saliva. Were they shedding virus? And then the antibodies is to ask, did they actually see the virus at some point? And as we move forward, we find that there's a pretty low infection rate in the community. About 2% of people who have enrolled out of the thousands and thousands that we've tested have had an infection. So in a sense, that's unfortunate. We're not closer to herd immunity. It would have been kind of nice if we had found that almost everyone were infected and we were kind of like almost done with all of this. But unfortunately, I don't think that's the case. I'll go a little bit more into the drug discovery. For me, I think career-wise, this was a very interesting time because I got interested in virology because of the possibility of viruses and infectious disease to basically disrupt society. And I was always fascinated about outbreaks like the Ebola outbreaks in the 70s. And I was kind of inspired to get into viruses because of this. So being a postdoc, I'm kind of in a place in my career where I can study a lot of different things at once. I have a lot of flexibility. So being able to do drug discovery and basic research questions on this virus in real time as it's outbreaking has been a really interesting experience. I'll just show a couple of basic slides. When we first got the virus, we weren't really able to, you have to do virology, you have to grow the virus. To study a virus, you have to have cells in a test tube, you have to add virus, and you have to measure how much the virus is growing. And one way you can do this is by looking at how much the virus is able to kill cells. So these cells here, these are called viro cells. They're actually monkey cells that the virus likes to infect a lot. So these are cells that I did not infect. You can see kind of the borders between the cells. These are healthy cells. They're very happy. This is 48 hours after I infected it. But then in the infected cells that you can see here, you can see kind of clearance. The cells are kind of missing from this spot. And you see these like lighter cells, they're kind of balling up and dying. And then if I look the next day, the uninfected cells are still pretty happy looking. And then you can see the virus just like burning through the cells, just killing everything. And this is essentially what's happening inside your lung. The virus is just killing your cells. So this is one way we can measure the virus in the lab. I'll skip that. And one thing we have to do is we have to figure out cells that we want to grow the virus in. And you can measure the virus over time. So this is a viral titer assay, where you can measure the amount of virus in infected cells over time. So as the bars go up, we have more virus. And these types of graphs let us know which cells we should use and at what points we should look for a given virus. And this is the amount of virus that we added. So we can measure this over time. And do this for a couple of different cell lines. And we can screen for drugs. So this is an example of actual drug screening that we do. We take these giant 384-well plates, so you can test 384 different conditions on one given plate, these small little conditions, and you can look for virus. So the color corresponds with how much the virus has grown. So if it's red, it means that the virus has killed the cell. So red is bad, red is dead. So on the left, these are control wells that were not infected, and they're light because these cells are still alive. And on the right, these are control wells that were infected with the virus. So if it's red, this corresponds to a drug that did not work. The virus still grew and killed the cell. But if you have something like this one right here, this would suggest a drug that worked really well to control the virus. And the shade of the color corresponds to how well it worked. So these gray ones are maybe these intermediate candidates, while the white one is a really strong candidate. And we've been screening hundreds and hundreds, actually thousands of drugs. So the FDA, the Food and Drug Administration, has libraries of drugs that are already approved for patients. So we've been testing these approved drug lists with the idea that if we find something, it can immediately go into the clinic and be used to treat people that are infected. So at the moment, we're in the process of writing up the drugs that we found, and we're trying to team up with biotechnology companies and clinicians here in the Bay Area to use these drugs in the clinic to affect change in COVID. So vaccines, this has actually moved a lot faster than you would ever expect for a vaccine. Generally speaking, vaccines have to move through a pipeline. They have to be, first you have to demonstrate that it works in the lab. You have to either use animals and protect them from the virus, or show that the antibodies produced in a human are protective. And then you can move into the clinic, and you can use small cohorts of patients for phase 1 clinical trials, where basically they're usually healthy patients that are volunteers, and you vaccinate them, and then you see how well they respond over time. If it passes that and it's safe, then it moves into phase 2. And then phase 2 is essentially a bigger study that takes into account a lot more variability than phase 1, which would be very small. And then phase 3, which is the hardest one, takes into account all sorts of folks. So generally phase 2 is larger, but they're usually healthy people, healthy young people who have strong immune systems who can respond well to vaccines. When you get to phase 3, you start enrolling children and you start enrolling elderly, people who may not have developed immune systems. So vaccines, to get through all of these pipelines, they generally speaking have to be able to work in everybody, and they have to be completely safe. And one thing that has always been kind of a concern for SARS coronavirus vaccines is a phenomenon known as antibody-dependent enhancement. where the antibodies you produce against the virus can actually make your infection worse. And other viruses like dengue virus are known to do this, but we don't know so much for SARS coronavirus 2. There's been a lot of studies that suggest both ways. But the bottom line is, originally we were anticipating, months to years before a vaccine was ready. There are hundreds of vaccines moving through the pipeline that are being supported by either companies or the government. And I think that there's enough that are in phase 3 right now that have done well that mass vaccination really could be achieved in the coming months. So it's been really impressive how that's moved forward. And this slide just kind of talks about different types of vaccines. More or less, you have to give some piece of the virus. So you can give RNA from the virus, you can give DNA from the virus, you can give a specific protein like spike I talked about earlier. You can have a harmless virus. So this is an adenovirus, which can be encoding pieces of the virus of interest, like SARS coronavirus 2, and you make an immune response against both this virus and the piece of SARS-CoV-2 that they're encoding. And then you can have inactivated virus. So you take real SARS coronavirus 2 and you destroy it, either through heat or some chemical, and then you give that to a person, or you can have live attenuated. There are no current live attenuated viruses for SARS coronavirus 2, so that's a little bit different. So I think I posed these questions in April. When does shelter in place end, and when does the SARS coronavirus 2 pandemic end? And the answers are unclear. The answers are that it depends on time. So shelter in place, actually a lot of places have opened up, right? So I think there's different levels of this. So the Bay Area is still under a shelter in place order. Most companies are working remote, schools are remote, but you can still go out and maybe spend time at the park if you're social distancing. I think that all of these will be, so it'll be community to community. It'll depend on current outbreaks. It'll depend on how many cases are going on. If things are too lax and cases spread, then generally speaking, we'll go back into a full shelter in place and restrictions will be tighter. If things continue to go well, things will be relaxed more and more. And there's always debates on what should you do. So people have said that one reason China's been able to do so well with it is because people are very respective of the authorities saying, okay, you got to stay inside. And in fact, it was enforced by police. You couldn't go outside. It wasn't an option. But I would say in the United States, at least, we're pretty individualistic. So people will do what they want to do. Maybe a lot of people are being, shelter in placing, but other people may not be. And as long as you have people not doing that, you're prolonging how long until we can completely clear it. And when does the SARS coronavirus 2 pandemic end? I'll say that even a vaccine, a mass distributed vaccine, will not end the pandemic. I think that the SARS coronavirus 2, and most virologists think that it's here to stay. It is part of our standard viruses that we have to deal with now. But the good news is it may not always be as virulent as it is now. It's not adapted to the human population, but it's staying with us, it's transmitting, and in a way it's selecting for people who are more resistant to the virus. The majority of people who get it are asymptomatic. So as time moves on, more likely, if you get infected by SARS coronavirus 2, it may not be severe. And even now, the majority of people don't get as severe cases, but it's the severity and the death rate being 1% to 2% is still extraordinarily high. But I think that what'll happen is this is just a virus that will be part of the human population. And within hundreds to thousands of years, this will become one of the common cold coronaviruses. And those common cold coronaviruses may have emerged like SARS coronavirus 2. But this is essentially part of our human history now. So these are some members of the Harris lab. This is Dr. Eva Harris at UC Berkeley, and these are some of the scientists in Eva's lab who do some of the work with me working on the virus. So yeah, thank you so much for your time. And I think Mitchell had mentioned that he had some questions that you guys had asked.
Dr. Buehring, thank you. That was fascinating. Just learned a ton just watching, there's, thank you so much. Paula, do you have anything? Yeah, I don't know if Paula hasn't said anything yet, everyone. This is my colleague, Paula, at the Conservancy. You've probably seen her on a number of recordings, especially if you check out our website.
Good. Good to know the recordings are being used.
So yeah, I have a few pre-recorded questions that students submitted, so I can just go ahead. Oh, yep.
Before you do, I would like Dr. Beering to talk about career path, because a lot of people probably believe that they would never be able to do what he does. And I just, a short biography, if you could, about how you got where you are.
Yeah, I would say that I think to do something like this, you just have to be interested in it. You just have to be generally interested. I had no biology background going into undergrad besides, biology classes in high school. And I think I went into college and I was just extremely interested by viruses and things that cause disease. And the first thing I did when I went to college was I wanted to go into a lab and learn how science was done. I had no idea about anything. I just what I did at first was I did a program called Work Study at UCLA, where essentially you can get paid to work in different environments. And I found a virology lab and there was a work study position available there. So I went into the lab and I did things like wash dishes and make solutions. And I was just so happy to be around science, and I would ask a ton of questions and, what is that? What are you doing? And, they were kind of like maybe ignorant questions at first, but I knew nothing what anything was. And then eventually, I learned enough in my classes that, I could kind of like talk about like basic biology. And they offered me like an undergraduate research position. I was working under my advisor there. His name was Hector, who just basically liked, I guess, excitement and like, oh, I wanted to teach you something. So I would just like basically like, lap it up, teach me anything you can, any experiment, just like from the ground up, right? And then after that, I worked with Hector for a while, and then he became a full professor at Washington State University. So then I went with him there and helped him start his lab. And I was trying to decide, oh, do I want to go to grad school? Because if you want to go into biomedical research and study things like infectious disease, generally speaking, you need a graduate degree, usually a PhD, which is a research degree, learn how to research. So I went with Hector and I wanted to see, could I do research full time? Because at that point, it was like it was a job in college, and I did other things as well. And then I tried it full time. I couldn't imagine doing anything different. I was just like enthralled with my question. I was basically paid to think about this problem and focus and obsess and just solve it. That's your job. And it's good because academia is a lot of flexibility in this sense. It's your job to creatively think and solve a problem. There's not generally hours. You don't have like a 9-to-5 type of thing. It's generally speaking when you're productive. So some people, they work really, really late at night. Some people work really, really early in the morning. Some people they transition. But going back into it, I went to grad school and studied viruses more and did more and more virology, and still loved it. And if you finish grad school and you're still interested in an academic like virology career, you do what's known as a postdoctoral scholarship position, which is more or less a transition phase to becoming a professor. So a PhD, I think you can think of it as a license to do research. It's essentially proof that you can do the entire research process, and you're advised by academics and professors. And when they approve your thesis, they're saying, okay, this person went through the entire research process. They know how to do appropriate controls. They know how to do research. And then the postdoc is your time to prove that you can do research. And then the transition here is you make a niche in the field. You ask some question that's really important. So one of the things I've been hoping to do with my COVID research is find a niche within emerging diseases with the flaviviruses that I study, like dengue, and then also mix that with a virus like coronavirus, because there are overlaps. Does that answer the question?
Yeah, it does. So some of the key things that I would would probably want to talk to a student who's looking at, maybe I want to go into a career in science or something, would be have curiosity, ask a lot of questions, find a mentor, and invest a lot of creative energy into into the career of being a student of science. Would you say that was correct?
Absolutely. Absolutely agree with that.
And the high school that you went to doesn't necessarily mean that you're going to end up going to, that you'll be able to do this or not. Isn't that correct?
Yeah, you can start from anywhere. I went to Fraser Mountain High School in Fraser Park, which I would say doesn't have an amazing track record for college prep. It does all right, but like, no, you don't have to go to a prep school. You don't have to come from money or like be taking a million AP classes. You can start anywhere, and just the energy and the desire to do it will drive you.
Yeah, and I've always said as an educator that it's curiosity that actually makes for a successful student.
Mm-hmm.
Thanks.
Yeah, thank you. I would definitely echo everything you just said, Dr. Beering. Thus far in my career, I majored in biology as well, and tried to get a lot of initial research experience as an undergraduate. And I was a pre-med during my first 3 years. I made it through all the prereqs of pre-med before I decided I didn't want to do it. And a lot of folks who are considering a career in science, they tend to look for, and I'm speaking to students, they tend to look for a very linear path. In medicine, for example, is relative to other disciplines, other professions. It's quite linear. You go to undergraduate, you get research experience, try to get clinical experience, then you do well on the MCAT, then you get perfect grades, and it's like very prescribed. And then you get into medical school, and you go through your medical school, you do your residency or you do your rotations. It's all, and eventually you get your own practice, or whatever the case may be, depending on which residency you get. It's very prescribed. That's just in the case of medicine. If you're entering into the research world, like Dr. Beering said, you pick up a lot of things piecemeal as you go. I can say in the plant world, where I do a lot of my stuff, the things I know, I picked up, some things from this person, some things from that person, some of the things I learned myself, some of it is trial and error. It's not like it's been A, B, C, D. It's been A, X, G, L, Y, T, all over the place. So, yeah. Yeah, thanks for sharing your experiences, Dr. Beering.
Absolutely.
With that, I have just about 10 questions from students. I'll go ahead and bring that up. 1 second here. Close that down. All right. Share, and where are you? There you are. Okay, so this was an assignment. Are you seeing the slide, or?
Yeah, I can see it.
Okay, you see my mouse moving? I have split screen. You can see that okay?
I don't see your mouse moving. It looks like I'm seeing your next slide view.
Okay, let me just remove the, okay. Let's try one more time, sorry. Stop. Okay, one more time. Sorry for the delay. And there we go. Now you should be seeing it.
Yep, that's good.
Okay, perfect. So this was called the Ask a Virologist assignment. Like I said, I just did it a couple of days ago. So I'm thankful for those students who submitted questions. You get 5 extra credit points. Thanks for doing it. Obviously, students, you didn't get a chance to watch Dr. Beering's presentation before you asked your question. So if your question has already been answered, that's wonderful. We'll skip over it. That doesn't mean your question was not valuable. It just means it's already been answered. So let's just go ahead. There's about 10 of you who submitted it. So first one was Alexis. And actually, technical thing here. I don't think I hit the to share computer sound button. Okay. Slowly but surely we will get there. Mm-hmm. Okay, so if you don't hear this, let me know.
Hello, my question is there any evidence showing that the COVID-19 virus is mutating at this time? and why or why not?
Did you pick up on that?
Yeah, totally. It's a very important question, and I think based off kind of what I was talking about, it gets at type of viruses this is. Coronaviruses are very unique viruses. They're RNA viruses, as I mentioned, and another general rule of RNA viruses is that they mutate a lot. But coronaviruses are a little different in the sense that they mutate a lot less than other RNA viruses, and the reason for this is they have a proofreading activity. So essentially what a mutation in a genome is when you're copying nucleotides into RNA or DNA, you put the wrong one. It's a different nucleotide, which can result in a mutation in a protein sequence. So what humans and what mammals and most eukaryotes have is a proofreading activity. So you have something in common that can sense when a wrong nucleotide has been put in. Most viruses don't have this. Coronaviruses do have this. So the question is it mutating? Yes, it's mutating because all viruses mutate, which means they adapt. From one person to the next, the virus looks different. But I think you may be asking the question, is it changing its biology? Is it getting more virulent? Is it getting less virulent? What's exactly happening? I think your question could be answered on a micro and a macro scale. On a macro scale, yes, of course, it's adapting. It's not used to humans, it's getting used to humans, it's changing. Naturally what will happen is the virus that is less virulent will be selected for, because a virus that is more virulent will kill the host and that virus may not transmit. But if a virus is able to keep the host alive longer and transmit more, that virus gets selected for. So there doesn't appear to be a lot of mutation that is making the virus more virulent. But yes, there are definitely mutations that are adapting the virus to the human population. So for us, that might be a good thing more than a bad thing.
Mm-hmm.
But at the end of the day, we don't know which directions things are going. Everything is still up in the air.
Yeah, I really liked how on your slides you mentioned you put your presentation together back in April and how much things have changed even since then.
Absolutely.
It's a few short months.
Science doesn't usually move that quickly. It really pushes it to the limit. Science is trial and error. And that's, I think, important to know that scientists don't know. And actually, scientists are wrong more than they're right, which is kind of like what makes us able to figure things out, because we don't mind being wrong. You know, you go with what you would expect, and then you find out if you're wrong or right. And if you're wrong, then you adapt your hypothesis. So at the beginning of this SARS coronavirus pandemic, virologists were telling a lot of people that, oh, you have gastrointestinal discomfort. That's probably not coronavirus, because coronaviruses are respiratory viruses. And we were all wrong. They are respiratory viruses, but they also go to different parts of your body. And this has been kind of what we've been finding out as we go.
I just read an article that was reporting on a study that was released that the spike protein has mutated to a gamma protein, which may allow the spike to attach to cells more easily, therefore being more contagious. I don't believe that's the same as virulent, but have you heard about that at all?
I don't know the specific studies. The other thing too, the literature is huge. But yes, there are mutations in spike that have been recorded that have basically replaced the virus. I don't think there's a lot of evidence on what those mutations actually do. So epidemiologists certainly record that viruses, for example, in San Francisco may look different than ones in LA. And over time, what is currently circulating is going to be replaced. There are now known and recorded mutations in spike that do certain things. Of course, spike is one of the surface proteins that's going to mutate the most because it's interacting with the host cell. But as far as how that changes its transmissibility and its virulence, I think is certainly unclear. And most studies that are done in cell culture, in vitro, so not in a human or not in an animal, they don't necessarily recapitulate what happens in a person. So for example, a spike protein, and I'm sure this paper is based off of something in cell culture, this mutant may attach better to cells, but actually in a person, that may be a bad thing for the virus. It may kind of raise your immune system's alarm too quickly.
Oh, interesting.
That's really interesting. Well, that's great question, Alexis. Let's move on to the next one. Okay, Alondra.
Question is on how the vaccine would make you less contagious of the virus. Like, how does it fight it off, or how does it like turn off the virus, or how does it minimize it?
Did you pick up on that? It was a bit quieter.
Yeah, no, I got that. So the question essentially is, how does a vaccine protect you against SARS coronavirus 2? How would that work? I think I'll answer this by kind of explaining what protective immunity is. There's a couple different definitions of protective immunity for vaccines. One can be considered sterilizing immunity, where the virus is just totally gone. It's cleared from your body. And the other one is containing the disease. But the way vaccines work and the way your immune system works is it has two major responses. So your body sees a virus, it sees an antigen, a protein from a virus, and it starts to make what's known as an innate immune response. It produces these things called cytokines, like I was telling you about, where it starts to bring in immune cells. And the two major immune cells are white blood cells, which I think are what they're commonly called, come in two flavors known as B cells and T cells. B cells produce antibodies. Antibodies are these proteins that will float around your blood, and they will bind to things that they're supposed to target. So if you get a vaccine against the coronavirus, you're going to make antibodies against coronavirus proteins like the spike. These antibodies will bind to the spike and they'll block it from doing virus things like entering cells. That's one way you can control the coronavirus, by producing these antibodies from your B cells. The other one are these T cells. And what T cells can do is they can kill cells that are currently infected with the virus. So maybe the virus is hanging out inside your cell. It's not floating around your blood where the antibodies are. It can still be detected by T cells, which will then kill that cell. So you have your B cells killing all the virus in the blood, and then you have your T cells destroying all the cells that it's hiding in. And together, that could produce sterilizing immunity and basically prevent you from being contagious.
Cool. Great question, Alondra. Next one here, Annabelle.
Since there's talks about attempting to get a COVID vaccine out by the end of the year, how long would it take for a typical vaccine to develop?
Well, I think we've already covered that one in some detail. Do you have anything extra to add, or?
I would just think that the normal scale of vaccine production is years. Given that things are successful, you have to assume that things aren't necessarily successful. And here we're going so fast because of redundancy. We have so many people trying so many different things at once.
Yeah, I didn't know there were hundreds going down the pipeline. That's a lot.
Absolutely.
Probably a record breaker.
Definitely. There's some concern to get this taken care of.
Okay. Next
Since there's
question. Okay.
Hello. I would like to know if someone has already had and recovered from COVID-19, could their body's natural immune response be enough to prevent them from being reinfected? And would they still need to be vaccinated?
Yeah, this is a great question. So the question is, if I heard it correctly, is if somebody gets infected with SARS coronavirus 2, do they have protective immunity? Are they protected from the virus? And if so, would they still need to be vaccinated when the vaccine comes around? So the answer is, I'm not sure. There had been certainly we know that people have protective immunity. Some people are totally protected from the virus, but there also have been confirmed cases of waning immunity and reinfection. So both situations happen. What we don't know is how common each one is. Have we been detecting exceptions, or have we been detecting the trend? Not enough people have been infected. Not enough. There aren't enough numbers to know which one is real. There's also a trend called waning immunity, where your body, when you get a vaccine, you make B and T cells, as I was mentioning, and they become memory, so they stick around for a long time. But your body will only keep them around if they're being used in a way, which is why you have to be vaccinated against certain things over time, or get boosters, stuff like that. So is there waning immunity against SARS coronavirus 2? Well, we've only known about this virus since more or less January is when we really started studying it. So that's 9 months.
Yeah.
Immunity is a 2-3 year process. So we won't know the answers to these questions for years to come. I imagine as far as public health goes now, the policy probably would be to vaccinate everybody, whether or not you can prove protective immunity. But it also could be a thing where if you're going to go out and do certain activities, maybe you have to prove protective immunity. Vaccines aren't necessarily going to give you protective immunity. You have to show you have antibodies over time. So unfortunately, there is no specific answer to your question. You need more time to really know for sure.
And adding on to that, wasn't it only maybe a month or two ago that there was the first positive case of someone being reinfected or having been diagnosed twice?
Yeah.
The first time early in the breakout, I think it was an individual in Arizona that got it early on and then got it later on. Definitely two separate cases.
Yeah. There's been several reports of these. There's a ton of caveats to these reports. Are these normal people? Are these people that have problems with their immune system? But yeah, certainly this virus has proven that it can reinfect. And we don't know if that's because you didn't produce an immune response in the first place. I will say that working with human samples, we've seen that people who have more severe disease appear to have a stronger immune response against the virus. So if you got a severe case, you're probably more likely to be protected than if you had an asymptomatic case. We find that a lot of asymptomatic cases don't produce lasting immunity. So if you weren't unfortunately getting really sick, you may not want to assume that you're protected.
That's fascinating. So much complexity.
Yeah.
Okay. Eduardo. There we go.
So I understand that vaccines contain pieces or parts of bacteria or viruses that they're trying to fight, but I just want to know what's actually in vaccines.
Yeah, that's a great question. We talked about it a little bit, but essentially it depends on the vaccine. So we kind of talked about the virus is made up of RNA, it's got proteins, it's got lipids. So some vaccines are the entire virus that's killed or inactivated in some way. Some vaccines are just pieces of the protein. And as I mentioned, there's a lot of vaccines that are in the pipeline right now. The most successful ones are what are known as mRNA vaccines. So they are basically the RNA of the virus, but mRNA essentially is messenger RNA. It means that it can be immediately translated into a protein. So you're both seeing the viral RNA, but then you're also seeing the viral protein because it's translating. So you're kind of seeing both. Vaccines usually have what's known as an adjuvant in them. I don't know if you've heard of an adjuvant, but an adjuvant is some compound that turns your immune system on. So in general, when you get infected by a pathogen, you make an immune response because it has antigens like proteins that you can make an immune response against, and it has a general danger signal that turns on your immune response. Oftentimes vaccines lack that, so you have to add in something that turns on your immune system. So a vaccine is generally an adjuvant and then a piece of the microbe, if not the whole microbe itself.
Awesome. And just to students, I know we haven't covered a lot of the concepts that Dr. Beering has been talking about in this class so far. We've just started on biochemistry for the microbiology unit. So we will, if you're watching this recording and we haven't gone over RNA, DNA, lipids, carbohydrates, the various biological macromolecules, we will go over those in some detail in some succeeding lectures. So that would be a good time to come back and watch Dr. Beering's presentation as well. So, and yes, excellent. Thank you so much. I think there's a couple more.
So,
Okay.
Is there any advantage to using advanced oxidation products in regards to killing or neutralizing viruses and bacteria? Or bacteria?
What was the product he said?
Advanced oxidation products?
It's like a cleaning product?
I suppose, yeah, advanced surfactants and things like that. And you did, yeah, I think that's what the question is pertaining to.
I guess I would wonder if what does it mean advanced? Does it mean stronger, like industrial grade? Or are we talking, yeah. I think that to answer your question, I don't think you need something crazier than like soap water and hand sanitizer. The virus is not hard to kill. I was mentioning that this is an envelope virus. It has a lipid on the outside, which means it's not very stable. So hygiene is important. Consistent hand washing or hand sanitizer like ethanol, alcohols are needed, but you don't need industrial grade or things that are super strong. And at the beginning, I kept getting, people would say, my grandparents wash our vegetables in hydrogen peroxide when they come. You don't need to do stuff like that. I think that doing things like not touching your face and being aware of what surfaces you're touching is enough. You don't need advanced materials. It's not a hard virus to kill.
Hi, so I want to know, what is the most effective type
Sorry.
of mask to use to further protect us from COVID-19?
I'm going to replay that. The question was, what's the most effective type of mask?
Hi, so I wanted to know, what is the most effective type of mask to use to further protect us from COVID-19?
I love it, a mask question. Definitely, this has evolved as the outbreak has progressed. At the beginning, people were saying things like, "Oh, you should wear an N95," and an N95 is a grade of mask that essentially doesn't allow particles smaller than a certain size to move through, and an N95 would exclude particles the size of viruses. So I guess by definition, that's the safest. But one thing we've found over time is that, I've said this before, the virus isn't very stable in the environment. It's actually harder to get infected from another person than you think. What is required is prolonged exposure to high doses. So a person who is shedding virus, if you're in close quarters with them for a prolonged period of time, that will increase your chances of getting infected. But for example, walking by someone on the street, if you wear like a normal face mask like this is going to be completely protective in that environment. I think that if you are indoors with somebody in close proximity, probably you're gonna have a much higher chance of getting it even wearing an N95. So the name of the game, I think, is yes, you can cover your face, you can wear a cloth mask, that's fine. There's types of masks that are vented. If they vent from the side, that's a good thing. But if they vent from the front, that's not a good thing. So some N95s, which are used for like painting and like chemicals, they vent in the front. That is actually completely not protective because you're blowing through, you're not taking it in, but you're blowing out. And the general rule is that the mask protects the people around you. It doesn't protect you so much. So it just keeps your breath in and any viral particles that you're shedding. So I don't have a specific type of mask I would recommend. I would say that just make sure your mouth and your nose are covered and that it's not vented in the front. But if you're maintaining social distance and not having prolonged exposure to people indoors that aren't part of like your household, you're probably going to be fine.
Mm-hmm. Mask wearing is like wearing a seatbelt for somebody else.
Right.
Okay.
So we've covered the basics on what COVID-19 is. I guess my question is, what exactly is it, going into detail? What is it like the test that they perform?
What do they search for exactly? Because I know they take blood and I know they do a swab test. What exactly in detail is COVID-19?
So we've covered a lot of that already, but anything other nuance?
Yeah, I think Cassandra's kind of getting into the diagnostic question a little bit. I mean, I think just to reiterate, COVID-19 is a disease caused by the virus, SARS coronavirus 2. When you get tested, they can either test, so blood tests for antibodies. Basically, it asks the question, not are you infected, but were you infected? Essentially, to make antibodies, you had to have seen the virus at least 2 weeks ago. It takes 2 weeks to make antibodies. So that's asking, have you ever seen it? When you take a swab test, I think almost 100% of labs right now are doing a PCR test, which means they are amplifying the RNA, the genetic material of the virus. And it's really specific, and it can tell you exactly, do you have virus shedding in your saliva? So you can spit and you can take a nasal swab. The nasal swab is usually higher shedding in your nose than there is when you spit. The minority of cases will be using a test called an ELISA, which tests directly for viral protein in a plate. PCR tests for the genetic material, and an ELISA tests for the protein. Most labs will be doing the PCR at this point in time.
In class, we'll talk about PCR next unit. Botanist that I am, I do know a little bit about PCR. So yeah, we'll go through an example of that. So yay! All right, a couple more questions.
Hello, I'm just wondering if the United States is currently manufacturing or just creating their own like self-made viruses. Thank you.
So self-made viruses.
I'm not exactly sure I understand the question. Is your interpretation to be that as like a scientific thing, like to study, or?
Perhaps, kind of, yeah, scientifically in the context of a virology lab. Yeah, that's my interpretation as well.
I will say that virologists are kind of like record keepers in a way. Anytime we see a new strain from a hospital, we like to sequence it and ask what its genetic material looks like so we can compare them to each other. And you can ask if these genetic differences have any biological differences. Labs in general will have many different isolates. I wouldn't necessarily call it stockpiling, but just you can ask these biological questions. So I think as we were getting at earlier, if something emerges and changes and all of a sudden diseases in humans are different, you would want to know why. And having viruses that we saw previously would help answer that question. So scientifically in that sense, but in the, I guess in like a bio warfare, like no.
No.
Stockpiling.
Okay, and the last question.
Hello, I have a question for the virologist. I was a nurse in the emergency department for the first 5 months of the coronavirus. I'm currently working for the county health department, and my wife is also a nurse. So we understand that the current precautions that are being asked of us are basically droplet precautions. So how long does the coronavirus last on a surface after the surface has been exposed?
Yeah, this is a great question. We've actually done a lot of studies in my lab on this specifically. One of the projects we were doing up front was we were working with a couple of different companies to determine what was the minimum you needed to do to PPE, personal protective equipment, to totally decontaminate it. And this was highly relevant to hospital workers because you would maybe be working and interacting with a patient that is infected. You're getting aerosolized droplets on your PPE that contain virus. So we were asking questions of how long does it stay on there and what do you have to do to decontaminate it? So the specific answer, of course, it depends on the amount of virus. we find that the virus, if in very low amounts, is extraordinarily unstable and doesn't prolong. Patients that are more sick early on are probably shedding more virus, so they would have more virus in that sense. When the droplet's dry and it desiccates, it's extremely bad for the virus, so that makes it extraordinarily unstable. Healthcare workers have the unfortunate challenge of working with and being in close proximity to the virus for long periods of time to kind of increases your chance because you're potentially being exposed to more and more. So maybe if somebody walks in off the street and maybe the droplet is dry for 2 hours, there's probably a pretty low risk of transmission. But if you're constantly, you know, in an environment where you're exposed to that, it could increase the chances. But I would say that within like a 2 to 3 hour window after desiccation, there probably isn't much virus left.
Yeah. My mom is a recently retired labor and delivery nurse. So the last 5 months of her career was navigating, you know, pregnant mothers who are in labor who are positive for the disease, and how to isolate that from infants, you know, newborns. And so that was, golly, had a huge list of protocols to follow. So
tough work.
That was tough work. Yeah, she retired. Anyway, Dr. Beering, thank you. That was just, I learned a ton. I know you'd mentioned previously that, you know, when your schedule allows, you may do a quick video of your lab space and show samples and techniques, whatever it is you're able to share, that would be very welcome. Yeah, just thanks again. This was great. I'll post this probably in a couple of weeks. So students, you're seeing this a couple of weeks later. But other than that, I look forward to chatting more.
That's great. Thank you.
All right. And